A warm summer day. The perfect day for a dragonfly to fly out. Or wait a second. Oh, that looks bad. Oh no. Oh, watch out. No. But what can scientists learn from such a single specimen and just a little drop of water?
But why do we collect these specimens? Just for the fun of it? No. The purpose of these specimens is to use them for research. Research towards understanding and describing Earth's biodiversity.
And we have quite some cool machines to take a better look.
This is the microscope for stacking photography. And this system is merging several photographs of the object at different focal planes and then create one picture which is totally in focus.
This is our field emission scanning electron microscope, which gives pictures of high resolution of the outside of the material. After the material has been coated, you can observe it with the microscope. For instance, here you see the wing of a dragonfly. You can see the hairs and the venation.
This is the transmission electron microscope. Here you can see the structures in a cell, and we are now watching at the pigment vesicles of a dragonfly. That is what gives the dragonfly a color.
This is the X-ray scanner for scanning in 3D in very high resolution. Now we're going to scan a dragonfly captures in the wild, especially the head, to see the outer structure and the internal structure.
Let's continue with the molecular stuff.
Thank you. So one leg of this insect is sufficient material for a DNA extraction. This extraction robot will perform the binding, washing and elution from the DNA. The rest of the DNA extracts will go in the DNA bank in the -80 freezer and can later be used for other research purposes.
So now we are going to submit our own sequence to the BOLT database and see whether there are any hits. The BOLT database brings together millions of barcodes and makes them available to scientists all over the world. There's our results. There are some very similar sequences, all of them belonging to the genus Drepanostichta.
With phylogenetic analyses, we can find out the evolutionary history and relationships among species. This is done by comparing genetic distances, taking into account substitution models. Naturalis has set up a high-performance computing environment in which we can run the necessary analyses. So now we can see the unknown specimen matches with Drepanostichta hermes.
Back to the water sample.
The ion torrent can generate millions of sequences in a short period of time. This allows us to analyze the DNA what was left behind in this water.
So here we analyze the data file from the ion torrent on the bioinformatics pipeline. We compare the data with the GenBank and with the BOLT database. And what we can see is that 30% of the sequences are from a dragonfly, but we also find surprisingly 6% of the sequences are Homo sapiens, probably Kevin himself.
The knowledge we have gained within this research project is not kept to ourselves. It is disseminated to researchers worldwide via scientific publications and to the general public, for instance in a museum.
But this was just a brief introduction into our labs. Let us help facilitate your future discoveries.